All right, so as Bob just said, I'd like to thank you for coming out on this cold and blustery morning straight out of a Winnie the Pooh story. So I just completed my first year as a faculty member here at Georgia Tech. And that first year has really been a wonderful experience for me, in part because Georgia Tech is remarkably multidisciplinary. It supports multidisciplinary research. And as you can see here, so this is my primary appointment here in the School of Biology, but I'm also an adjunct member in the School of Chemistry, a member of IBB, as well as a member, very active member of a number of centers. These aren't just trivial memberships or paper memberships. These are centers whose goals directly align with research themes in my laboratory. And so this multidisciplinary supportive atmosphere is certainly reflective also in the composition of members of my research group, where I have 9 people currently, and they're all doing different things. So I have molecular evolutionists, chemists, molecular biologists, computational scientists, and globally, they're all contributing to a common theme. But individually, they're all doing separate things that don't have much overlap. Okay, so globally, what we're trying to do is develop something that we have termed in evolutionary synthetic biology. And we want this to be more than just a simple catchphrase. We really are trying to turn this into a paradigm, so to speak, a way to do research, where on the one hand, we want to use evolutionary principles and models to develop engineered proteins as well as potentially engineered organisms. On the other hand, we want to generate synthetic biomolecules and use these to enhance our understanding of evolutionary principles or structure-function relationships of biomolecules. And so there's this circular relationship here where at the heart we have this interest in structure function, and our ability to understand structure function is driven by evolutionary theory, which, as you know, is a descriptive or explanatory science. And on the flip side, we have this interest in biomedicine and biotechnology, which is obviously an applied or a translational science. So we want to be able to take research themes over here and apply them to structure function analyses that help us develop evolutionary theories, as well as take known standard evolutionary theories to understand structure function relationships that help us develop novel biomolecules. So at the heart of what we do are sequence analyses, biological sequence analyses. And so here we have what my PhD advisor likes to say is nothing more and nothing less than a simple linear string of chemicals, okay? But our ability to extract information out of that linear string of chemicals is what's important for our laboratory, okay? Specifically, we're trying to find some sort of information that tells us about the function of this protein, or maybe where this protein came from a historical or evolutionary perspective. And so you can imagine, though, that by itself, this really isn't very informative, right? And it's not until you can put the sequences within a comparative structure does this allow us to extract information that is useful, okay? So what we would do then is we would collect what are called homologous sequences, okay? These are sequences that share a common ancestor. And you collect as many sequences as you can, and it's not really until you collect enough sequences that you are able to extract some sort of useful information, okay? So for example, in this one, maybe someone already extracted something from there, but you can see, you can highlight some sort of information within a linear string of chemicals, so to speak, okay? Now, just because you can extract or highlight some sort of information doesn't necessarily tell you what its function is or where it came from, okay? You just know that each one of those 3 species has a gene, and that same gene believes that this is a true statement, but you don't really know why that gene maybe feels, believes that way, so to speak, right? So what we want to do is we typically put these sequences within some sort of historical context. And from an evolutionary perspective, what this means is we try to analyze these sequences within a phylogenetic framework. And the phylogeny then allows us to analyze sequences within an evolutionary history based on extant organisms to understand their shared or common inheritance. Okay, so we can ask which extant species evolved from the same ancestor, and how were ancestral traits modified in different lineages. And so for those of you maybe that are coming from chemistry or from biomolecular engineering, this is a common way to represent relationships within biological sequences. Okay, so this is the so-called phylogenetic tree, where in this tree we would have extant organisms represented by these nodes here, what are called terminal nodes or leaves or tips of the tree. Okay? And these terminal or modern or extant sequences will share common ancestors at different points in the tree. So here we have these two modern organisms share a common ancestor here. These three modern organisms share a common ancestor here, and then there's the common ancestor for all 5 sequences or organisms on this tree. And they're related by edges of the tree called branches. Okay? So this really forms the heart of what we try to do. We take biological sequences, we put them in a phylogenetic context, and we try to extract information within that context. Okay, so just to give you a brief example of how phylogenetics can be important, there's the classic case of an example where a doctor had injected his mistress with a needle that had HIV-positive blood in it. Okay? And so this was from 1994 in Louisiana. And a woman claimed that her ex-lover injected her with HIV blood, and the records had shown that, in fact, the physician had drawn blood from an HIV patient that same day that the lover claimed that her physician lover had injected her. And so we want to be able to prove or demonstrate that the blood was from an HIV patient ended up in the woman. Okay, so how do we do that? So some background information about HIV is we know that HIV has a high mutation rate, which can be used to trace paths of transmission or evolutionary paths associated with the evolution of with those gene sequences. And so this assumes then that 2 people who got the virus from 2 different people will have 2 very different HIV sequences. And so what we can do is we can try to build phylogenies. Okay, we can build these phylogenetic trees based on HIV sequences to be able to attempt to extract something useful from these sequences. And so what people did was they took multiple samples from patients, the woman and controls, and they built these phylogenetic trees. And what they noticed is that there's this so-called nesting of the victim sequences within the patient sequences that the doctor had extracted HIV from, indicated that the direction of transmission was, in fact, from patient to victim. And so this was the first time that phylogenetic analysis was used in a court case. Okay? And so here's the tree, all right, where we have this green cluster was the HIV patient that the doctor was treating, and here we have the sequence from the ex-lover. And as you see here, this is an expanded analysis of the initial analysis. As you see here, that all of the victims or the ex-lover sequences clade or fall in or nest within the single patient's HIV sequences.
What role do the other sequences play? Are they necessary for establishing the directionality, or could this have been done with just these 2?
It could not be done with just these 2. You need directionality, it demonstrates, and it also demonstrates the probability of chance alone in 2 sequences being related. Okay? All right, so that was just to demonstrate how important a phylogenetic analysis can be and how potentially maybe we can use a phylogenetic analysis. So let me now talk about more specifically what we do with a phylogenetic analysis and how we can use that to extract particular information that's useful for our lab. So there's this concept in molecular evolution called rate heterogeneity. Okay? And one form of rate heterogeneity is called heterotachy. And what rate heterogeneity allows for is unequal mutation rates among sequence positions within a collection of aligned sequences. Right? And specifically, given that pattern, you can have something called heterotachy. And what this model allows for is the mutation rate at positions to vary in different branches of the evolutionary tree. All right. So that doesn't make sense probably yet. So let me explain exactly what this means. So rate heterogeneity, here's an example of rate heterogeneity. We have a single gene, let's say, and we have homologs from 4 different species. Okay? And what you see here is that the first 2 positions are slowly evolving. Okay? They don't change at all throughout the evolutionary history of these 4 species. What you notice for the third position, you see a moderate amount of mutation or evolutionary change. Okay? And that's evident by here you have a conserved hydrophobic property associated with these amino acids. And so for the 4th position, again, it's conserved. You don't see any change here. And the 5th position is rapidly evolving. Okay? It doesn't seem like there's really any selective constraint on what amino acid is occupying that position. And so if we were to schematically represent that, we would show this here where we have site 1 just has a single circle in it or a single ball to represent a low evolutionary rate. Same with position 2. Position 3, however, has a moderate rate represented by 2 balls. Site 4, a single ball, and the last site is rapidly evolving, so it has the most balls in it. So the way that evolution is typically modeled up until my PhD work is that you assume this process was homogeneous, okay? And what that means is, let's say here, again, we have this collection of sequences. Okay, so this is not a single sequence. Let's say this is the last common ancestor of life, and this is represents a gene within that population of, say, tens of thousands of individuals. And you're looking at the mutation rate at the individual sites within this gene in the population. So the first site is slowly evolving, the second site is moderately evolving, the third site is slowly evolving, and the 4th site is rapidly evolving. What we had typically assumed is that this process stayed the same throughout time. So as the last common ancestor of all of life diverged to give rise to bacteria and eukaryotes, those individual mutation rates stayed the same throughout time. So the first site remained slowly evolving in both bacteria and eukaryotes, and the last site remained rapidly evolving in both bacteria and eukaryotes. And so what my work showed was that, well, in fact, this is not a homogeneous process. Rather, this can change throughout time. And so you start with this example again here where we have the last common ancestor. We have the same distribution of mutation rates. But what happens now is as bacteria evolve as they split from eukaryotes, the first site goes from being slowly evolving to now being rapidly evolving. Okay? Conversely, the 4th site goes from being rapidly evolving to now being slowly evolving. And so we've seen this shift then in rates, and presumably that shift is somehow related to a functional constraint that's acting on that gene and as it differs between bacteria and eukaryotes. So this gene presumably is doing some small minor changes in its functionality or its properties between these 2 groups of organisms. So what happens though is that it can be difficult to detect this signal because what we typically do in the field of molecular evolution is we just identify a whole bunch of sequences, we throw them together, we build multiple sequence alignments, we build phylogenetic trees from those multiple sequence alignments. And so since there's no bifurcation or dichotomy in the analysis of those sequences, we can often miss these patterns. Okay, so if this pattern existed, normally what we do is we just take a bunch of bacterial sequences and a bunch of eukaryotic sequences, throw them together into a multiple sequence alignment, and if we were to look at the evolutionary rate of those sites, we would not see that bacteria is rapidly evolving at that position, whereas eukaryotes is slowly evolving at that position. Rather, that position would just look like it's moderately evolving. Okay? So I spent my PhD career developing statistical tools to be able to identify this pattern here. Okay? And so let me just walk you through then an example of the functional, how that pattern is manifested at the protein level or the functional level. Okay? So here's an example where we have a particular site. This site is slowly evolving in bacteria. Okay, so we have a conserved arginine protein for the chemists, biochemists, and molecular biologists in the room, whereas this same site is rapidly evolving in eukaryotes. Okay? So it suggests then that there's a strong selective constraint acting on this site in bacteria, but that this constraint has been lifted or removed within eukaryotes. Okay? So when we compare a particular gene family, in this case something called EFTU, between bacteria and eukaryotes, here's an example of the distribution of these types of what are called heterotachous sites, sites that have a shift in their evolutionary rate between bacteria and eukaryotes. So shown in green are the sites that are rapidly evolving in bacteria, but they're slowly evolving in eukaryotes. Conversely, the sites in red are rapidly evolving in eukaryotes, but slowly evolving in bacteria. Okay? So we distribute these sites onto a known three-dimensional structure of the protein, and what we tried to do is be able to make functional inferences or functional explanations as to how these patterns can arise. Okay? So let me give you a brief background of exactly what this gene family does. As I mentioned, it's a gene family called EFTU, and it's a G protein involved in translation. Specifically, what EFTU does is it brings amino-acylated tRNAs to the ribosome. So EFTU protein will bring all amino-acylated tRNAs to the ribosome. When the correct anti-codon-codon base pairing takes place between the tRNA and the messenger RNA, that causes hydrolysis of the GTP molecule. That EFTU and the spent nucleotide are then released from the ribosome. Okay? Then something called a nucleotide exchange factor in bacteria, this is represented by EFTS. This nucleotide exchange factor kicks out the spent nucleotide and allows for a fresh activated nucleotide to rebind the EFTU molecule. Okay, so what we knew at the time is we had a structure for EFTU. We did not have a structure for the homolog in eukaryotes, but we also knew where the nucleotide exchange factor bound onto EFTU. Okay? So what we wanted to do is be able to make these explanations, functional explanations as to what's going on with this distribution. And so you can see here, for example, we have this collection, large collection of red sites that are in this area here. And this is where we knew that the nucleotide exchange factor bound EFTU. Okay? So these red sites, remember these red sites are rapidly evolving in eukaryotes, but they're slowly evolving in bacteria. So we inferred then if they're slowly evolving in bacteria, and we know that they are interacting with the nucleotide exchange factor, but these same sites are rapidly evolving in eukaryotes, then it suggests that the nucleotide exchange factor is not binding in the same way in bacteria as it is in eukaryotes. Okay? And so we made this prediction. We made additional predictions as well. We predicted that this area here, so these are, remember now, these are green sites. These green sites, these are rapidly evolving in bacteria, but slowly evolving in eukaryotes. Okay? We predicted that this area here would actually be an alpha helix in eukaryotes. We predicted that this is probably a binding domain in eukaryotes because these sites here form a hydrophobic conserved region in eukaryotes, but these sites are rapidly evolving in bacteria, which is what you'd expect for something on the surface of a protein. So if something's on the surface of a protein and it's not really doing anything in terms of binding ancillary proteins, then it's probably more rapidly evolving. But in eukaryotes, these sites are slowly evolving. And so, sure enough, when our paper was in press, somebody had determined the crystal structure for the eukaryotic factor. And our predictions turned out to be correct. This region ended up being an alpha helix. And this region is where the nucleotide exchange factor binds in eukaryotes, okay? So a completely different binding region for the nucleotide exchange factor, even though these blue proteins are performing the same function within bacteria and eukaryotes, they have a completely different way of doing that function. Okay, so what's never been demonstrated up till this point, though, is nobody's ever experimentally validated this concept. Okay, so this is what we are currently doing in the laboratory. We are trying to take these sites, mutate specific sites to be able to engineer these elongation factors from binding their nucleotide exchange factors between the two different domains of life, the bacteria and the eukaryotes. Okay? So I'm not going to show you any results because we don't have any results, but I'm just going to set the stage really so that in the future, when we talk about this or someone from my lab presents this, you'll at least have some background information as to where they're coming from. And so here we have the nucleotide exchange factors between bacteria and eukaryotes. These are now the known structures of these. So in red here is the EFTU in bacteria. And in eukaryotes, unfortunately, they don't call it EFTU. They call it something else, EEF1A. Okay, and that's what's shown in cyan here. And then we have the nucleotide exchange factors for bacteria shown in green and for eukaryotes shown in yellow. And so what we want to do is we want to identify these heterotachous sites that I talked about previously, and we want to see if we can swap out heterotachous sites between bacteria and eukaryotes. Okay? So there's going to be sites. There are going to be sites that are conserved in bacteria that allow the EFTU to interact with the nucleotide exchange factor EFTS. Those same sites, since they don't do anything over here, those same sites are going to be rapidly evolving. Okay? So what we want to do is we want to take the site from bacteria and put it into eukaryotes. And we'll do this for a whole slew of sites, and with the intentions then that we can get this bacterial EFTU, this red protein, to bind to this yellow protein, or the nucleotide exchange factor from eukaryotes. Okay? So we want binding partners to swap with the fewest or the minimal number of mutations possible, where those mutations are driven or dictated or highlighted based on this evolutionary analysis. Okay? And so here we can align, this just demonstrates that we can align, structurally align EFTU and EEF1A. And so these are the sites that we will be mutating in EFTU in our attempt to get them to bind, to get EFTU to bind the nucleotide exchange factor from eukaryotes. Okay? So these are all heterotachous sites. These are sites that are rapidly evolving in bacteria, but slowly evolving in eukaryotes. And so we are changing these sites in bacteria with the intentions or hopes that they will allow then the EFTU to bind to the yellow protein. Okay? Conversely, we're doing the same thing in bacteria, or the EFTU, in the EEF1A in eukaryotes, where we're mutating that protein to be able to get it to bind to the green bacterial nucleotide exchange factor EFTS. Okay, and here are the distribution of heterotachous sites in this example here. Okay? So this is really just setting the stage then for hopefully what are exciting experimental results that we will be generating in our laboratory shortly. So let me shift gears then. I'm going to try to shift gears a lot here today. As I mentioned, I have 9 different research projects in my lab, and I don't have time to talk about all those 9. But it's not going to keep me from trying to talk about all those 9. So another thing that we are interested in is being able to engineer or exploit the protein translation system in an attempt to develop therapeutics or novel proteins that contain unnatural amino acids. Okay? So this is a research theme that's of particular interest to the therapeutic community or the biomedical community, because proteins that have unnatural amino acids have a lot of therapeutic value. So what we want to do is we want to manipulate specific interactions within the protein translation system, and those are interactions between transfer RNAs and elongation factors that bring those transfer RNAs to the ribosome. And so this historical view, though, of tRNAs is that they are interchangeable components and passive adapters of protein translation. And there's this new view, though, championed in particular by Uhlenbeck at Northwestern, that states that no, rather tRNAs are active molecules adapted to meet specific kinetic requirements of protein translation. Specifically, what that means is that the cognate 20 tRNA amino acid pairs have similar thermodynamic contributions when they bind to EFTU. And so when you misacylate, you put the wrong amino acid onto a tRNA, it can have a broad spectrum of thermodynamic contributions to EFTU binding. And so what Uhlenbeck's laboratory did was they essentially mischarged a whole bunch of tRNAs with a whole bunch of different amino acids. And then they looked at those mischarged tRNAs and their ability to interact with EFTU. And what they found was that there is this dichotomy between whether or not EFTU recognizes the amino acid or whether or not it recognizes the tRNA. And that this relationship is inverse. So if you have a tRNA, okay, so here let's take the glutamate tRNA. If that tRNA interacts strongly with EFTU, then the amino acid group on that tRNA, the glutamate itself, that does not interact with the EFTU molecule. Okay? Conversely, if you have an amino acid on the tRNA that interacts strongly with the EFTU, so glutamine in this case, that tRNA does not interact strongly with the EFTU. Okay? And what this allows then is that a particular thermodynamic compensation between the tRNA, the amino acid, and their interactions with EFTU. All right? So if you were to take a weak amino acid and charge it onto a weak tRNA, then presumably that mischarged tRNA does not bind strongly enough to EFTU in order for that charged tRNA to be delivered to the ribosome. So what that means then is if we were trying to make proteins that incorporated or contained unnatural amino acids, as a first order, what we'd want to do is we would want to bind unnatural amino acids to these so-called strong tRNAs, because presumably EFTU can't bind strongly to an unnatural amino acid. So if we stick that unnatural amino acid onto a strong tRNA, then the strong tRNA should be able to deliver that to the ribosome. Okay, conversely, what we can do is bind unnatural amino acids to weak tRNAs and simply hope that the EFTU molecule can bind that unnatural amino acid with similar thermodynamic affinity as the cognate amino acid. And so you can imagine, though, that there's limited combinations of non-cognate tRNA amino acid pairs. So a higher order rule, it would be we want to interconvert a weak tRNA into a strong tRNA. Okay? And this would presumably allow us then to stick anything onto any tRNA. All right? And so what we want to do is we want to be able to exploit this observation that Uhlenbeck noted, is that there's this GU base pair present in the weakest as well as the strongest tRNAs, but they really haven't established any specific rule as to the distribution of that GU base pair in the tRNA molecule. So what we want to do is exploit our evolutionary models to determine sequence structure-function relationships of tRNAs and their interactions with EFTU. So specifically, this GU base pair, as I mentioned, what it does is it generates or presents an increased electronegative potential to anything that is juxtaposed on the EFTU molecule to interact with that increased electronegative surface. And so what that could mean then is if you had a positively charged amino acid on EFTU, it could interact more strongly with this GU base pair than, say, it would with a GC base pair. Okay? And so what we did was we analyzed tRNA sequences, and we have this, again, here's this phylogenetic tree, or this cladogram that represents relationships of the sequences. And this clustering is based on the distribution of the GU base pair. And what we noticed is that the so-called strong binding tRNAs have a GU in a specific place on the structure of the tRNA, whereas the weak binders have a GU in a different area of the tRNA. Okay? So specifically, you have when the GU is around here in the acceptor stem, those are the strong binding tRNAs, whereas the weak binding tRNAs have a GU in the T stem. Oops, sorry. So what we want to do then is mutate individual sites based on our evolutionary analysis. So here's an example that we are working on. We have a tRNA for valine. Okay, valine is one of these weak tRNAs. So what we want to do is convert this weak tRNA into a strong binding tRNA. Okay, so here is the GU. So the GU is in the T stem. It's flipped. This orientation is flipped from the previous slide. We're now looking at the mirror image of the tRNA. So here we have this GU, and what we're doing is we're mutating not only the GU, but we're also introducing a GU into the acceptor stem, because that's where we notice GUs happen to be for these strong tRNAs. So in this example, we've taken the valine tRNA, the weak binding tRNA. We're introducing a GU in the acceptor stem up here, as well as right here. And we also have examples where we are getting rid of the GU in the T stem. Okay? And so the way that we're going to measure this is we're using something called the PURE system, which is a reconstituted in vitro translation system, where you isolate all of the individual components of protein translation, except for the ribosome. You isolate that from an organism. So recombinantly purify, express all of the individual protein components of the translation system. In this case, we're using E. coli components, and this is a collaboration we have with Matt Hartman, where we can then manipulate the system. Okay, so instead of putting in, say, wild-type tRNAs, we can put in our engineered tRNAs and look at the amount of protein that's being made in this system. Okay, so again, here we're working with the valine tRNA, which is a weak tRNA. And we're not yet working with unnatural amino acids. What we're going to do to test our idea is stick different natural amino acids onto this valine tRNA. So we're going to stick a weak amino acid and a so-called strong amino acid onto this weak tRNA. So this combination of the valine tRNA with the glutamate weak amino acid, you wouldn't suspect that this would participate a lot in protein translation. Okay? Conversely, you would expect this pair to participate a lot in protein translation, because now here you have a strong amino acid with a weak tRNA. And so here is an example. This was this is our first assays that we've done so far. And what we have here are two groups. We have the example here where we take the glutamate and we stuck it onto the valine tRNA using a ribozyme. And then we take the glutamine amino acid and stick it on the valine tRNA. And so you can, although these are preliminary results, you can identify some general patterns that are occurring here, okay? So when we take the wild-type valine tRNA and stick a weak binding glutamate amino acid onto there, this doesn't participate a lot in full-length protein product, okay? Conversely, when you start making these mutations into the valine tRNA, okay, so here, whether you're making it in the acceptor stem, the T stem, or in both, what you notice is that there's this increase in this ability of the mutated tRNA to participate in protein translation. So if you just get rid of the GU in the in the T stem, what happens is that molecule can then participate more strongly in protein translation. And the combination of getting rid of the GU in the T stem as well as adding the GU in the acceptor stem allows for this charged tRNA to participate really strongly in protein translation. Okay? Conversely, you would expect in this case, where you have a strong amino acid and a weak tRNA, is that it really shouldn't matter what the mutations are in that thing. Okay? And besides this example here where we know we have some error, you see in general that you have this strong amount of participation regardless of what the mutation is. Okay? So what we're doing is we're doing a lot more examples where we put different amino acids onto different mutated or engineered tRNAs, ultimately with the intention is that we can get these things, these engineered tRNAs, we can stick unnatural amino acids onto them, and then they will be able to make proteins that contain unnatural amino acids for us. Okay, so let me shift gears again here and now talk about this concept of ancestral sequence reconstruction that we work on in the laboratory and how we are exploiting this research theme to allow us to better understand evolutionary principles and also allow us to develop novel biomolecules. So here is a very brief history of Earth. As maybe some of you know, Earth formed about 4.6 billion years ago. And for roughly the first 700 million years of our planet's existence, we were being bombarded heavily by planetesimals that would have certainly vaporized any liquid water present on the planet. And so shortly after this heavy bombardment ends, we know that the Earth's crust solidifies, the oceans form, and then sometime after this, 3.9 billion years ago, it's controversial, but at some point, certainly after that, you have this evidence or biosignatures of life. Okay? A lot of these are contentious. People argue over them. Probably one of the least arguable examples of a signature of life left in the rock record is this example here, where we have a roughly 3.5 billion year old rock from Western Australia, and you can see these patterns in the rock here. And it's believed that these patterns are created by organisms like this that currently live on our planet in Australia and the Caribbean. These are, for those of you that don't know, stromatolites. They are essentially very large biofilms where hundreds, thousands of different species are living together in these clumps. Okay? And so what we want to know is, or what we are interested in is, what kind of environment do these ancient organisms live in? Okay? And so the field of molecular evolution offers a clue in terms of the structure of the so-called tree of life, where here we have the tree of life with the three domains of life, bacteria, archaea, and eukaryote. And this is roughly believed to be the overall distribution of life on that tree. And shown in red then, the red branches are, these are what are called hyperthermophilic organisms. Okay? Hyperthermophiles are defined as organisms that grow at or above 80 degrees Celsius. Okay? So these things love living in hot environments. Conversely, that would be compared to organisms called thermophiles, which grow between 45 and 75 degrees Celsius. And then you have mesophiles and psychrophiles, which grow at even lower temperatures. So, but based on the distribution of these hyperthermophiles on the tree here, it's believed that the last common ancestor of life, or the last common ancestor of bacteria, also lived at a hyperthermophilic environment, or a very, very hot environment, such as nowadays you can find these things called black smokers or hydrothermal vents on the ocean floor. Okay? So typically, this notion of understanding ancient environments is conducted, researched by geologists. Okay? So what we want to do is we want to be able to pretend like we are geologists and go back in time where instead of looking at a rock, we want to look at sequences, biological sequences, and try to extract some information from those biological sequences that give us a clue as to an ancient environment. Okay? So the analogy that we typically talk about in our lab is that of what a historical linguist does. So a historical linguist will look at the relationships and spellings of a modern word. So here we have the modern word for snow. And based on this distribution of the modern word, you can estimate what an ancestral spelling of the word snow is, or an ancestral pronunciation of the word snow. So in this case here, we can go back 5,000 years to the Proto-Indo-Europeans that gave rise to these modern languages. But in this case, the Reconstruction says something about the Proto-Indo-Europeans, right, in their environment. Specifically, they lived where it snowed. So we want to be able to do the same thing, but with biological sequences instead of man-made words. Okay, so we're going to have this transition, essentially going from the human alphabet to nature's alphabet. We use that same thing that a historical linguist does, the same type of analysis, but use biological sequences, place them within a historical context or an evolutionary tree, and then estimate what the ancestral spelling of that gene would be. Okay? So in this case, then, we would just replace languages and words with a particular gene, and we would estimate what that gene sequence would be at particular nodes in that phylogeny. So we are reconstructing the ancient sequence associated with this gene. Okay? In our case, we're interested in something that goes back to about more than 3 billion years ago with the dawn of bacteria. So what we have then is we have this phylogenetic analysis. Okay? This phylogenetic analysis allows us to infer the ancient sequences at the individual nodes here. Okay? This is all computational. And then we actually build those sequences in the laboratory. We recombinantly express those genes, make the ancient protein in the laboratory, and measure some sort of function associated with that ancient gene. Okay? Again, I'm still working with this elongation factor TU gene family. It's turned out to be wonderful for our lab. And what's nice about this gene is that it essentially serves as a molecular thermometer. So the gene, the protein, creates a thermostable protein when that protein is isolated from thermophilic organisms. But whereas that protein is not thermostable when it's isolated from mesophilic organisms. Conversely, the EFTU from thermophiles is not optimally functional at mesophilic temperatures. Okay? And so this means there's this linear relationship between optimal binding temperature and optimal growth temperature of the host organism that has that protein. Okay? And so, for example, we have a visiting scientist in my lab, Michael Gromia, who's the world's expert in structural bioinformatics, who's already demonstrated this relationship with other proteins. Okay? There's this near linear correlation coefficient between the optimal temperature of a protein and the host temperature, okay, of the organism. Okay, so here we have this thermometer. So what we want to do is we want to be able to look at the specific thermostability profile of the proteins, whether they're modern or ancient, to give a clue as to what type of environment those proteins come from. So here's an example. This is just to show you this relationship in modern organisms. Here we have the E. coli protein. We are using circular dichroism to look at the stability of that protein. And so as those of you that work with E. coli, you know that it grows optimally at about 40 degrees Celsius. And so the melting temperature with the EFTU protein is right around the optimal growth temperature of its host organism. Okay, the same thing with the protein from Thermus aquaticus. The optimal growth temperature of Thermus is around 75 degrees. The melting temperature of the EF protein from that organism is around 77 degrees. Okay? So what we did was we generated a phylogeny. Okay, here's these trees again. And we computationally reconstruct ancient sequences at nodes of this tree. And then we build these sequences in the laboratory, recombinantly express those genes, and measure the thermostability of those ancient proteins. And what you notice here, this general trend, is that as you go further back into time, whether you are starting out with these modern organisms or whether you are starting with these modern organisms, the further back in time you go, the more thermostable the ancient protein is. Okay? And so that allowed us to correlate this temperature trend to known geological evidence for the ancient ocean temperatures. So here we have an estimation of the ancient ocean temperature based on the fractionation of oxygen isotopes in the rock record. So if we go back 3.5 billion years ago, the ancient ocean is predicted to be very hot. This is degrees Celsius here on the y-axis. Okay, so 80 degrees Celsius is extremely hot for us, at least. Okay, and as you approach modern day, the present time, you have this near linear decrease in the temperature of the ancient ocean. Okay, here's another geological-based study looking at a different isotope, and you see the same general trend. And so what we were able to do is we were able to put time points associated with our ancient proteins, where here we have our tree again, and we have different nodes that we reconstructed on this tree, and on the axis down here is we have time. So the blue dots represent all the different nodes that we reconstructed in ancient EFTU, and then we have the time associated with those divergence dates, those nodes. Okay, so that allows us to, we have temperature now, we have time now, so we can plot our results onto this same type of graph. And what we notice then is this same general trend, whereas very ancient life lived in a hot type of environment, or at least the ancient life that gave rise to modern life lived in a hot environment, and those organisms progressively adapted to their changing environments, as would to be expected under the paradigm of Darwinian evolution. Okay, so let me then just spend the last couple minutes here talking about how we are extending this work. Okay, so one thing that we are trying to do is be able to use this information to understand how thermostability evolves in proteins. Specifically, we want to know if we can exploit this information to build models that attempt to engineer thermostability in different protein families. And this is, in a lot of ways, this would be kind of the holy grail for biotechnology and industrial processes, because often these processes require catalysis at elevated temperatures for enhanced product substrate solubility, viscosity, and catalysis, as well as to prohibit biological contamination. So if we are able to develop an algorithm or a model that allows any protein to become more thermostable than it currently is, then that would hold terrific value for those communities. And so what we are doing right now is we are looking at the structures of these ancient EFTU proteins, a collaboration with Eric Ortland, a crystallographer at Emory, and work with Michael Gromi in my laboratory, where they are crystallized. So here is an example of aligned ancient EFTU structures that we've crystallized and determined the three-dimensional structures. And so what we want to do is we want to be able to use this information to help us develop models where what's interesting in this case, so we have, let's say, an ancient protein here, an ancient protein here. We know the amino acid changes that occur between these two nodes on the tree. And we want to be able to predict which of those changes are responsible for that change in thermostability associated with those ancient proteins. Ultimately, being able to take multiple paths along this phylogeny and look at these structures and make predictions, and then actually test these predictions in the laboratory and to make a database that could be exploited by others with different interests. Another thing that we are trying to do is we're trying to replay this so-called tape of life. This is based on this notion, famous evolutionist named Stephen Jay Gould at Harvard, who made this comment that any replay of the tape of life would lead evolution down a pathway radically different than the road actually, he didn't really say that, rather than the road actually taken if we could replay the tape of life, okay? So what we're doing is we are taking these ancient sequences, these ancient genes, this ancient EFTU, and we are sticking it into a modern organism where we've gotten rid of the endogenous protein, okay? So of all the thousands of genes in E. coli, we can knock out one of those genes, stick in the ancient one, and watch it evolve in the organism, okay? And what we know is that there's going to be this strong selective pressure for this ancient EFTU molecule to accumulate mutations to change its thermostability profile when it's evolving and interacting within the E. coli organism. Okay? In addition, we're trying to address this criticism where, how do you know you've made the right ancestral protein, right? We don't actually know unless we can talk to God, unless we can time travel. We don't know what the true ancient sequences are. And so what we want to do is we want to be able to benchmark ancestral sequence reconstruction. And so this is what Ryan Randall in my laboratory is doing, where we are taking a red fluorescent protein, okay, and we are randomly mutating it, and we are selecting for variants that have particular phenotypes. And so what you can potentially imagine then is we can take this sequence, we can evolve it in the lab, we can evolve it in a way that is similar to how evolution works. So we can evolve it along this tree-like structure. Okay? So we can mutate the sequence, we can look for variants. Say, for example, we can look for variants that maintain the same phenotype. We can look for variants that have new phenotypes. And at the end of the day, we're going to have a whole bunch of sequences. We're going to be able to take those sequences, plug them back into our computer algorithms that predict ancient sequences, and we're going to then rebuild the ancient sequence and see if we can recapitulate this green phenotype, this red phenotype, this red phenotype, and this red phenotype. So this allows us to benchmark ancestral sequence reconstruction algorithms. Okay? The ultimate ancestral sequence reconstruction would be not just a gene, not just a pathway, but a whole organism. Okay? And this is what Ziming Zhao is doing in my laboratory, where she's trying to computationally reconstruct a complete bacterial genome with the intentions of exploiting recent technological advances at the Venter Institute, where they have not only synthesized a complete bacterial genome, but they've also transplanted one bacterial genome into a different host species. And then that host is able to divide, and one of those daughters has the synthetic genome. And so this technology then is essentially ripe for us to be able to take advantage of to potentially create an ancient organism. Okay, so she's doing this with mycoplasma. Another research project in my lab, this is work being done by Megan Cole, and what it relates to is the origins of life. And so I like to, for those of you that don't know, Nick Hudd is Georgia Tech's resident expert in origins of life. And what I like to tease Nick Hudd, because there's this notion of the RNA world that predates the modern world, and that I like to say that the RNA world is so-called hollow at the core, because people are try to use RNA as the single biomolecule for life. Okay? And what that means then is that there's this potential paradox, because life as we know it today is a two-biopolymer system where you have information and catalysis being done by two different molecules. Here you have DNA, here you have protein. And so there's this paradox. How do you have just a single molecule acting as both the information content of life as well as the catalysis of life? Now, of course, we know that this happens in real life today in the form of ribozymes. But in the RNA world, we have this potential situation where maybe there never really was just RNA around without protein. Okay? So what we know is that based on interstellar clouds, meteorites, the Stanley Miller experiments, there are lots of amino acids around. Okay? And what we also know based on random RNA libraries is that you can identify ribozymes that catalyze peptide bond formation. You can identify random RNA molecules that can catalyze amino acylation of tRNA. So really what this does is it can argue for a scenario where you have these statistical random peptides just lying around in early life. And so you can ask potentially, well, is there a scenario where a particular type of pool of random peptides would have contributed to, in some advantageous way, to early life? And so that's what Megan Cole is working on in my laboratory, where she's trying to understand this transition from an RNA world to a ribonucleoprotein world. Okay, she's taken two approaches, where hopefully those two approaches will meet up in the middle with what we're doing with the Georgia Tech NASA Astrobiology Research Center. Okay, and as well we have Josh Stern, who's working on trying to resurrect a whole ancient ribosome, or the last common ancestor's ribosome. And this would be analogous then to say what NASA is interested in is this planetary exploration. Well, what we're doing in our center is the ribosome exploration. We're taking lots of different approaches really to understand how the ribosome has evolved. Okay? So lastly then is, so as I've shown here today, we have a lot of different research themes in my laboratory. Okay? And my students like to kiddingly argue as to which research is the most exciting. But the one research theme that has particular interest to me is a project that James Kratzer is working on, because my PhD, I was trained in a biomedical field, okay, but I applied molecular evolution to the biomedical field. So the ultimate would be to somehow generate a therapeutic potentially for biomedicine, but that is based on evolutionary analysis or evolutionary work. And so that's what James is currently doing. Unfortunately, we don't have patent protection on this yet, so I can't publicly disclose what we're doing since we're being video recorded here. But I would encourage you to corner James at some point and ask him what he is doing. And so with that kind of anticlimactic explanation there, I'm going to end and thank you for your time this morning.
Questions? So I have a quick question. It seems like a lot of what you're doing has a lot of implications for biotherapeutic manufacturing. You talked about the thermal stability. What about the engineered tRNAs? Is there, what's the implications for that on biomanufacturing of therapeutics?
Beyond just making proteins that have unnatural amino acids, we haven't given any consideration yet. But that's why I'm at Georgia Tech. So there's collaborations all over the place, and so hopefully that's an example that's right for the picking. Steve?
So looking at the superposition of EFTU and EF1A,
Yeah.
It doesn't look like some of the regions you're working at. It looks like the tertiary structures are actually substantially different. Possibly.
Well, they are, yeah, there are differences, okay, clearly. But so what we've done, though, is that we expect, so we have this, this sphere, this 5-angstrom sphere that rolls around and tries to, if you're within that sphere and you are a heterotachous site, then you will potentially contribute to this change in binding partners. But we can, the worst case scenario, you're right, is we introduce these changes, and the tertiary structures are so different that those sites are not going to be able to interact with the complementary nucleotide
My
tree.
question was whether you think we will actually shift the tertiary structure
Oh,
from one to the other.
oh,
In addition to
yeah,
the
right.
one
I don't know. I don't know what it takes to shift to the tertiary structure. But we are making different combinations of sites. So maybe we will be able to see this shift occur over time. And since we're collaborating with a crystallographer, maybe we can crystallize these things. Okay. Yes?
Are there common features to the thermal stability in terms of amino acid sequences?
Yeah, right. The short answer to that is no. Okay? People have been trying to identify some common property that you can exploit to convert a non-thermostable protein into a thermostable protein. They've been trying for years. No one has really been successful. We can hack at it a lot until we can get a protein to be thermostable, but it ends up not really following the same path that we set out to do. And so, but that's what we're trying to create. To be honest, I don't think we'll be all that successful, but I think we will make some contribution towards it that allows us to take a step in that direction. Roger?
Is there different rates of heterotachy within different bacterial phyla? I say that because the Thermophilic seem to be very close to the ancient EFTU on your cladogram, in the sense of which amino acid rates are changing a lot.
So are you asking about sub-heterotachy?
Yeah, within bacteria, because you said that you had examples between eukaryotes, for example, and bacteria.
Right. So we've never looked at heterotachy within a, say, domain of life. We've never looked for heterotachy within bacteria. Presumably it's there because there are major bifurcations within the bacterial domain. But no single, the way that the algorithms work, no single sequence is going to mess up our analysis. So if we had a completely conserved aspartate in all of bacteria, except Thermatoga had a tryptophan, that's not really going to affect our analysis because it takes branch lengths into consideration. Okay. Yeah, Sujin, last question.
Isn't there gene duplications going on in eukaryotic genes, and how do they influence? Maybe they have some complementary or redundant functions that.
Right. So not with this gene family. There's few examples. So elongation there are different elongation factors. Okay. And this family is not one of the ones that's experienced a lot of gene duplication. Okay, it's also the reason that we're able to exploit this family so much is that it's also one of these families that doesn't appear to be horizontally transferred at all, because that would also really screw up the analyses. But potentially you could, if you did have duplication events, as you know, right, you could have, if there's, say, subfunctionalization between those duplicates, then that would certainly obscure this signal of functional divergence. But in fact, in that case, you would be able to look at paralogs with this functional divergence technique and be able to identify the sites potentially that are responsible for the subfunctionalization. Okay? All right, so with that, why don't you get on with your days, and thank you for coming out this morning.